get started. It's 12:01, so it's a pleasure for me to be here to give you this renal ground rounds today. This is the first time I'm talking about this particular project. I know we have some experts in the field, so I appreciate your attendance and hopefully your questions. The title of my talk is Oxalate Toxicity, elucidating novel mechanism of calcium oxalate induced cell death in the intermedullary collecting duct. So today we're going to be talking exclusively about the intermedullary collecting duct. I have no disclosures. So I want to start with the most important slide first. And it's this slide that acknowledges the people that actually did the work. A lot of the work I'm going to show you today, the data, actually came from my lab, which is just right around the corner from this room. I want to acknowledge specific people, including Niharika Bala, a third-year doctoral student in my lab. She did a lot of this work. Dr. Rafay and Zainab, they both contributed a lot of their time and efforts to this project. Dr. Yu did all of the electrophysiology studies. I'll show you. Lauren and Dr. Dewan and other folks also contributed to various aspects of this project. But also I want to collaborate my my other team members, collaborators. Dr. Khan, I don't know if he's here, he was looking for the room, hopefully he found it, or he'll find it. He is a world expert, international expert in kidney stone research. He's won, I think, 3 Lifetime Achievement Awards. I don't know if anyone's winning more than one. He's won 3 here at UF. Also, Dr. Denslow, she's about to retire at the end of the month, but she's been a long-time collaborator. Dr. Bird, he and I have been collaborating for several years, and he just gave a renal grand rounds here on this topic, kidney stone research. A few weeks ago, Dr. Rupert Rishi is sitting here. She is another one of my valued collaborators. And of course, Dr. Kazori, we presented a poster at the ASN meeting on some of the data I'll show you today, but most of it's unpublished data. So I want to thank my team. This is my lab. I have a pretty big group. I can't mention everyone. I forgot to mention someone, and I apologize, but this, we have a theme, alligator. One of my students made that, but I want to thank everyone that participated in this project. So I want to start with the objectives, learning objectives for this lecture. The first one is to explain why recurrent kidney stone formation is indeed a big public health problem. Next is to discuss oxalate metabolism and how the kidney handles oxalate. The third is to explain oxalate toxicity in tubular epithelial cells. The next objective is to distinguish the correlation between different types of lipid classes, lipid species, and in the context of the pathophysiology of calcium oxalate kidney stone formation. And the next objective is to appreciate how crystals, calcium oxalate crystals, cause different forms of cell death, including apoptosis and ferroptosis in the intermedullary collecting duct. And the last objective is to discuss implications for lipid raft formation and activation of this NLRP3 inflammasome signaling pathway induced by calcium oxalate crystals in the intermedullary collecting duct. So we'll start with the problem, and that is recurrent kidney stone formation is a big public health problem. So the incidence of kidney stone formation it is increasing worldwide, not just in the U.S. And the underlying mechanisms of the process for this kidney stone formation and kidney tubular cell damage is not well understood. In fact, we have more questions than we do answers. Although we have been studying this for many years, we still have a lot of mechanistic questions to answer. So types of stones. And we heard this before from Dr. Rucci's grand rounds and Dr. Bird's grand rounds, so I won't spend too much time on that. But the most common type are these calcium-containing stones, calcium oxalate, or calcium phosphate, or a mix of both. And the second most common type are these uric acid stones. I won't talk about them today. we're focused primarily on calcium oxalate or crystal formation in this particular talk. So the process of kidney stone formation depends on an imbalance between excretion of water and insoluble stone forming salts, leading to a high concentration that supersaturates urine and the intermedullary collecting duct fluid. So, patients, one of the things that is common to do is tell them to hydrate, right? Dehydration is a big problem, and can be potential health just by hydrating. But in some cases, those stones need to be removed surgically, and Dr. Bird gave a whole beautiful grand rounds on how he does that, so I won't spend too much time on that. And you can't see it, but it's blocked by this thing here, but effective treatment is limited. So the three most common medications, and I'm not going to talk too much about them, I just state that the three most common medications for prescribed to kidney stone patients are thiazide and thiazide-like diuretics, potassium citrate, and arotherminal. So Dr. Gucci and Dr. Bird talked in depth about this in their renal grand rounds. So I won't talk too much about that. So this is a big question. And I consulted with Dr. Khan, who is now here, sitting in the back. He has done a lot of work in this field and was able to answer my question. So that's this question. What is the initial site of crystallization? Is it the interstitium? Is it the tubular lumen? Or is it the renal calyx? Well, if you're talking about animal models, most animal models, the answer would be it's the tubular lumen. And if you're talking about stones that form crystals that plug the collecting duct, then you're also talking about tubular lumen. But the most common calcium oxalate stones actually adhere to plaques plaques on the surface of the papillary, and those plaques start from calcium phosphate deposits, and that is in the interstitium around the loop of the Henle, and that grows through the interstitial connective tissue and then breaks through the papillary surface, and so you can see here from my diagram. Here we have renal plugs if we're talking about the collecting duct. And then we have these renal plaques that I just alluded to, and we have these deposits, these plaque deposits here, and those are initially made up of calcium phosphate. So that's the answer to this question. So oxalate, it's actually been in the literature a lot, but it was recognized in certain plants in the 18th century. And it wasn't until 1839 that oxalate crystals were identified in urine. And approximately 2/3 of kidney stones contain oxalate as the major constituent. And I want to point out two important studies that will be important for this project that I'm talking to you about today. And that is PLG and this Garcinia Cambodia extract, also known as hydroxycitric acid. So these two studies showed that these compounds were efficacious and effective in dissolving calcium oxalate crystals, and in part the PLG inhibited HIF-1 alpha-mediated inflammatory responses and oxidative stress damage associated with aggregation of these calcium oxalate crystals. So I'm going to base my premise, my rationale on these two studies for using these two compounds. There are a lot of compounds that we can evaluate, but these are the two I'm going to be talking about today. All right, so how we're going to talk about how oxalate is metabolized and how the kidney handles oxalate. So unlike the byproduct urea, which is actually a very useful metabolic waste product, and we actually need some of that waste product, especially important for concentration of urine. This oxalate absolutely useless. I can't think of any beneficial use of this oxalate metabolic end product. That's formed from the byproduct of the metabolism glycoxylate and ascorbate. It's formed and it's excreted into the urine where it poses a problem because of the insolubility of its calcium salt and promotion of calcium oxalate crystals in the urine. So oxalate results, and it's metabolized in the liver by lactate dehydrogenase 5 enzyme, isoenzyme, and then it's filtered and secreted in tubule. So it's secreted in a tubule by SLC26A1 and SLC26A6, and its concentration in the urine is relatively low, usually less than 0.5 millimoles per liter, but it can precipitate with calcium and form calcium oxalate crystals. So most patients with calcium oxalate stones have no disorder of oxalate metabolism, but there are 4 disorders which have been demonstrated to produce increased excretion of oxalate in humans, and they are here. I'm not going to talk too much about them, but excessive ingestion of oxalate itself or one of its precursors, and you have other disorders as well. But I just want to point these out. I'm not going to really focus on these today in my talk. Oxalate toxicity. So this is important. You'll see in the literature a lot of studies have studied oxalate toxicity in various renal epithelial cells, including human proximal tubule cells. But the thing is, crystals don't start from proximal tubule cells, and they don't attach well if they do. So these studies were done mainly to show toxicity and mechanisms, but we're using another cell type that's more relevant, and that's the medullary collecting duct cells. But this particular study I'm referring to, what they did is that they did a series of experiments to show that these proximal tubule cells, so LLC PK1 is porcine, pig proximal tubule cells, and these HK2 cells are human proximal tubule cells. So in both porcine and human proximal tubule cells, they were really sensitive to oxalate, and it induced the calcium oxalate crystals induced severe toxicity and cellular injury and death. And they concluded saying compared to, they also utilized the mouse intermedullary collecting duct cell, compared to that MIMCD3 cells, those cells are more resistant than the 2 types of proximal tubule cells to injury. But it doesn't mean that they're, not, that calcium oxalate is not toxic to the cells. And that's what we wanted to address in this study. So apoptotic injury promotes calcium oxalate crystal attachment in IMCD3 cells. This one study, Kleinman et al., they examined changes in the subcellular localization of this nucleolin-related protein expression during the course of cisplatin-induced apoptosis in these cells. And they found that pre-existing apoptotic injury promoted the calcium oxalate crystal attachment in an NRP-dependent manner. So they concluded by saying if the cell is already injured or damaged, that's going to exacerbate the calcium oxalate crystal attachment and effect. So next part of my talk that's important is I'm going to be talking about different types of lipids, and this is not new in the context of kidney stone research. So our very own Dr. Khan did a lot of seminal work in lipid biology and kidney stone research. And phospholipids play an important role in kidney stone formation, and Dr. Khan and his group explored the presence and dense abundance of various lipids in the urine of kidney stone formers, and also in the matrices of those actual kidney stone biopsy samples or kidney stone specimens. And they found that there was a significant increase in the amount of total phospholipids and in the urine of those kidney stone formers compared to healthy individuals. And also, kidney stone formers showed a significantly greater amount of phospholipids than those with uric acid stones or truvite stones. And among those phospholipids were sphingolipids, sphingomyelin, phosphatidylcholine, and phosphatidylserine. Now I'm going to focus, you're going to hear about these lipids in a minute. That's why I'm introducing them now. But they found significantly elevated concentrations of these lipids in the urine of these kidney stone formers. So phosphatidylserine eversion is an important process that happens in the early stages of kidney stone formation. And I included some of the important studies in terms of signaling cascade related to this concept. And the TGF-beta SMAD signaling pathway was shown to play a role in increasing phosphatidylserine. Also, Dr. Khan's group investigated how phosphatidylserine facilitates the nucleation of these calcium oxalate crystals. And also, exposure to oxalate itself was shown to cause a redistribution and externalization of phosphatidylserine. So these are some studies that motivated us to do a lipidomics study for the first time using these intermedullary collecting duct cells as we investigated potential mechanisms by how calcium oxalate could induce cell death, mitochondrial dysfunction, as you'll see today. So we have to consider lipid binding proteins, and some of the most important lipid binding proteins are these annexin family of proteins. And the clustering of phosphatidylserine membrane domain was shown to be induced by this annexin complex, annexin A2 S100 complex. And one study actually showed caffeine reduces crystal density, decrease intracellular calcium, and promotes translocation of annexin A1 to the apical plasma membrane. Another study, and this is relevant to Dr. Rucci's grand round, where she showed us that women are protected from developing calcium stones. And this could be part of that mechanism. Estrogen and protective effect against calcium oxalate kidney stone formation. So we have to consider this very important signaling mechanism if we are going to investigate mitochondrial dysfunction and cell death. And that's the piezo-1 NLRP3 signaling pathway. So my lab has been studying this pathway for a few years now. We have all the molecular tools and models to do so. So recent studies have shown the variant or overactivation of this NLRP3 inflammasome, and presumably from an increased density in lipid rafts. So lipid rafts are signaling domains. And we showed in other studies that when you have an increase in lipid raft formation, we have activation of this LRP3 inflammasome pathway. And this leads to various forms of cell death, including apoptosis, ferroptosis, necroptosis, pyroptosis, and other forms of cell death as well. So again, we're talking about these human proximal tubule HK2 cells. One study by Chen et al. Showed that HK2 cells in culture that were challenged with oxalate showed ROS involvement or dependent increase in the NLRP3 inflammasome complex. So the rationale for our study is that the NLRP3 inflammasome or activation of that NLRP3 inflammasome leads to the formation of cytosolic signaling complex that serves as a platform for caspase-1 activation associated cell death. So I just alluded to NLRP inflammasome is a complex, and part of that complex is a caspase-1 protein, and the caspase-1 is essential caspase regulatory protein that regulates other caspases, and that's important in cell death. So our first hypothesis was a mechanistic hypothesis. There is a mechanistic hypothesis in which calcium oxalate augments, increases phosphatidylserine membrane levels, also augments lipid raft density, and also activates piezo-1, leading to mitochondrial dysfunction, NLRP3 inflammasome activation, and cell death in medullary collecting duct cells. That's a mechanistic hypothesis that I'll show you data for in a few minutes. The next hypothesis is a more preclinical therapeutic application hypothesis, and that is engineered immunoliposomes that we make and load with various pharmacological inhibitors and drugs, selectively suppresses piezo-1 ion channel activity and inhibits overactivation of the NLRP3 inflammasomes, prevent mitochondrial dysfunction and cell death. So the experimental approach that we used. One, we used this these mouse cells, mouse intermedullary collecting duct cells, as our first model. And this is a widely used model. It actually recapitulates human cells very nicely because they endogenously express the urea transporter, A2, urea transporter A2, and this is important for urea recycling. They also express epithelial sodium channels, functional channels, and because of that, they generate current. And then finally, they express water channels, alpha-chloride 2 channels, which are important for water homeostasis. So this is a very good cell type and model cell to use for various reasons. So we do a lot of lipidomics in my lab, and I just want to make sure everyone is clear on the procedure, how lipidomics is done. So lipids are extracted from membrane fractions of these cells. I just alluded to that. Now, collecting duct cells by high-performance liquid chromatography that's coupled to a mass spectrometer equipped with electrospray ionization. Now, we're talking about using an amine column here. And next we have a mass spec that's actually a triple quadrupole mass spec. And I'm going to break down the different quadrupoles. So the first quadrupole is used for the measurement of the mass lipid. The second quadrupole involves nitrogen gas inducing a collision. The third quadrupole involves the separation of daughter ions. And because we're using an amine column that interacts with charged head groups of these lipids, we have the elution here in this order. So hydrophobic lipids are detected first. Hydrophobic lipids, the PAGs, the DAGs, the MAGs, the sphingomyelins, all these are hydrophobic. And then these are the negatively charged ions, or not ions, lipids. So phosphatidylserine, lysophosphatidylserine, phosphatidylinositol, lysophosphatidylnositol. These are all negatively charged. So lipid quantification is then performed. So as I alluded to earlier, mitochondrial dysfunction was a part of our goal and part of our hypothesis. So the gold standard to measure mitochondrial dysfunction is to use the C-Horse platform. So we do a lot of C-Horse in my lab. And basically it measures two metabolic pathways, mitochondrial respiration and glycolysis in live cells in real time, and provides functional kinetic measurements of cellular bioenergetics. So each injection here, indicated by the vertical down arrow, blocks different complex in the electron transport chain in the mitochondria. So here, the oligomyosin, we're blocking ATP synthesis. The SCCP, we're uncoupling phosphorylated oxidation, and this retinone antimyosin A, we're blocking complex 1 and 3, which completely shuts down the entire electron transport chain. So when we block ATP synthase using oligomyosin, we can actually gather information about ATP-linked respiration and proton link. And when we uncouple phosphorylated oxidation using FCCP, we can measure maximal respiration and also spare capacity. And last bullet here is upon inhibition of ATP synthesis by oligomyosin under an increased rate of glycolysis as a compensatory mechanism. So the next approach, I can't speak too much about it because I have a patent that's in process. So on this approach, but I'll tell you a little bit about it. So these are engineered liposomes, and they are engineered to target specific cell types, and they can be loaded with anything, any pharmacological inhibitor, any agonist of choice, any drug of choice. And they're specific for it's like a lock and key mechanism. They're specific for a particular cell type. So of course, the cell type that we're interested in this project are the intermedullary collecting duct cells. So what we did is that we fused an antibody against the UTA receptor on the surface of these liposomes. So that monoclonal antibody specifically binds to the UTA receptor expressed on the surface of these intermedullary collecting duct cells. And the next thing that we did is we conjugated these these are phosphatidylcholine liposomes that were conjugated to Texas Reds, who we can actually see where they're going. We can actually see them being taken up by the cells, and it's pretty cool. So that's pretty much all I can say about them. I'm allowed to say about them. But one more thing. We loaded them with PLG, and that's one of the two compounds I told you about earlier that have been shown to reduce oxalate toxicity, right? But we can load them with anything that we want. Okay, so now here comes the results. So rationale for doing lipidomics again is, this was motivated by Dr. Khan's beautiful studies. He publishes wonderful papers in high impact journals, including Kidney International, all the time. And every time I read one of his papers, it gives me an idea and motivates me to do something new. He actually recruited me to this field. I wasn't interested in kidney stone research before I met him. So he actually recruited me to the club, and now, I'm going to kidney stone meetings and doing this kind of research. So, thank you for that. But I can't talk about retirement. He's an emeritus professor, but enjoys being retired. So the rationale for studying these lipids are membrane lipids promote the formation of calcium oxalate, calcium phosphate containing kidney stones and become a part of their stone matrix. So this study that I'm referring to by Dr. Khan, this group, showed that urine of both calcium oxalate and uric acid stone formers contain significantly more cholesterol ester, and triglycerides in the urine compared to healthy subjects. But urine of calcium oxalate stone formers contain more acidic phospholipids. So this experiment was done 3 times, 3 independent times. And basically what we did is that we took these, cultured these mouse intermedullary collecting duct cells, and we challenged them with calcium oxalate for 24 hours. We saw that we did for different time points, but we didn't see any effect after 8 hours or 12 hours, but 24 hours was the magical number. So after 24 hours, we saw a change in various lipids. And if you look here at the PLC analysis, PTA analysis, you can see nice separation between the 2 groups. The green is vehicle, and pink is calcium oxalate. The volcano plot, we identified several lipids, including some bioactive lipids that were differentially up or down regulated in each of the 2 groups. And from this heat map, you can see, or maybe you can't see, so I'll read them out to you. so you can see the red, and this here, the green row on the top represents vehicle. The left part represents calcium oxalate treatment, and you can see here the red in the heat map is the lipids that were most significantly changed between the two groups. So for the calcium oxalate group, we have 1, 2, 3, 4, 5, 6 different phosphatidylserine residues that are significantly enriched in the membranes of these mouse and medullary collecting duct cells after calcium oxalate treatment. Again, these are representative of 3 independent experiments, and there's also, in addition to that, a PG, phosphatidylglycerol, and there's also a PI, phosphatidylinositol, actually 2 PIs, that could potentially be important. So next, because we're talking about lipids, we had to investigate lipid binding proteins. We looked at a lot of different lipid binding proteins, and one of them was the Nexin family of proteins, Nexin A2. And here in this particular experiment, we transfected these mouse intermedullary collecting duct cells with DS, red marks protein to label the inner leaflet of the plasma membrane, and also cytosolic marker. And after 48 hours of transient transfection, we treated these cells for 24 hours of calcium oxalate or vehicle. And then we did some immunofluorescent microscopy experiments, and you can see here again, this is representative of 5 different experiments. When you treat cells with calcium oxalate, that causes a rapid translocation of annexin A2 to the plasma membrane and or the cell surface compared to vehicle treatment. And the annexin A2 in this case is labeled with alexafluorofluoride green. The blue represents DAPI. So we have the same number of cells in each group. And then where you have yellow, that's colocalization of DS red marks with annexin A2. So next we measured whether calcium oxalate treatment can cause mitochondrial dysfunction in these cells. And lo and behold, it does. So calcium oxalate decreases non-mitochondrial respiration, decreases basal respiration, decreases sphere capacity, and decreases maximal respiration. The stars represent significant. 2 stars represents a p-value of less than 0.01, 3 stars less, p-value of less than 0.00. Okay, so now we want to, because we showed that the calcium oxalate crystal was causing mitochondrial dysfunction, we wanted to know whether or not it was also causing cell death, and what kind of cell death. So we have antibodies that serve as markers for different forms of cell death. For ferroptosis, we use this one antibody, anti-ACSL4, and for apoptosis, we use this antibody, anti-caspase-9. So indeed, calcium oxalate treated cells showed an increase in ACSL4 protein expression and an increase in caspase-9 protein expression. And these experiments were done 4 times, 4 or 5 times, 5 times maybe. And clearly you can see we have both apoptosis and ferroptosis happening in these cells after treating the cells with calcium oxalate for 24 hours. So because I told you that lipid RAS play an important role in the signaling microdomains that lead to the recruitment of other signaling proteins and activation of ion channels and inflammatory proteins that can be linked to cell death, we wanted to ask the question whether or not calcium oxalate could induce lipid raft formation in these cells. So we treated the cells with cholera toxin type B conjugated to alexafluorofluoride, and after pre-treatment with that cholera toxin B that labels lipid rafts, so the cholera toxin specifically labels lipid rafts, and if you have positive signal, it'll appear green. We also treated the cells with calcium oxalate for 24 hours or vehicle for 24 hours. And you can clearly see that cells treated with calcium oxalate had a lot more lipid raft formation density compared to the vehicle treated cells. And this was done several times. Experiment was done several times, very reproducible. So calcium oxalate is causing an increase in density in lipid rafts. Why is that important? That's important because lipid rafts have been previously shown by our group and other groups to activate piezo-1, an ion channel called piezo-1. And piezo-1 is important for many reasons. And one of the reasons is that it is associated as part of the NLRP3 signaling pathway. As I alluded to earlier, and as I'll show you in a few minutes. So piezo-1, this is the piezo-1 activity here measured by pap clamping, conductance of 20.2 picosiemens. Dr. New did this pap clamping study. The Harkabala did the mitochondrial dysfunctional studies. And you can see here piezo-1 activation with the agonist YODA. YODA-1 is the agonist for piezo-1. DMSO is the vehicle. We have a decrease in maximal respiration, decrease in basal respiration, decrease in ATP production. So then we investigated whether or not piezo-1 and the NLRP3 protein co-localized in lipid rafts in these cells. And CAV1 is a lipid raft protein, and you can see here, this is a sucrose gradient fraction. So the first 4 fractions are lipid raft fractions. And then the other fractions are various fractions associated with organelles of the cell, including the Golgi and ER and nuclei. Nuclei is 11, plasma membrane is 6, 7. But you can see here, piezo-1 and NLRP3 co-localized in the fraction 2, 3, and 4. Okay, so next we investigated whether calcium oxalate can increase the density of NLRP3 in these cells. And again, this is immunofluorescence microscopy, and we show here that indeed it does. The green is positive signal for NLRP3, but this is total NLRP3, not membrane expression. So then we corroborated those results by doing confocal microscopy, and you can see the Z-stack here, the vertical in the horizontal bars here. You can see NLRP3 labeled in green. This punctate staining indicates it's in the nuclei when you treat cells with vehicle. But when you treat cells with calcium oxalate, look what happens. There's a translocation in increase in membrane expression. You see the outline of the membrane. You can see here in the Z-stack of the NLRP3 protein. So NLRP3 membrane protein expression, not just total NLRP3 expression, but membrane expression also increased after calcium oxalate treatment. Okay, so now this is the last hypothesis we're trying to address, and we use that engineered immunoliposome strategy to load our PLG in these immunoliposomes that have a cell surface antibody against the urea transporter receptor. And you can see here, we looked at caspase-9 as our indicator of apoptotic cell death or apoptotic marker. Now, these cells were treated with calcium oxalate for 24 hours. But the first group, and this is 5 independent experiments that we loaded on the same gel. So the first 5 experiments are cells, MIMCD3 cells that were treated with calcium oxalate for 24 hours in the presence of these immunoliposomes that had that drug, PLG. And the last 5 are, they were treated with vehicle instead of the immunoliposomes. And you can see here from the densitometric analysis from the Western blot normalized to the Ponce stain that the immunoliposome significantly blunted the increase in caspase-9 protein expression, indicating that it was suppressing apoptotic injury. So this is specific for those IMCD3 cells. We actually did this in other cell types, and there was no effect. Presumably because those immunoliposomes were not taken out by those cells. They didn't have the UTA transporter. So again, it's like a lock and key mechanism, very applicable to cancer biology, but we're using, a hybrid form of it to treat cells that are important for kidney stone formation. So it brings me to my summary slide, and again, we'll finish on time, maybe a few minutes early, so we'll have time for questions. So the first point, hopefully I convince you that calcium oxalate increases the concentration of phosphatidylserine in membrane fractions of mouse intermedullary collecting duct cells. And annexin A2 is recruited to the plasma membrane after treatment of calcium oxalate. And we think it's a protective mechanism because annexin A2, I've been studying this protein since 2009. And it actually has protective roles in wound healing. So it's important for repair of the plasma membrane. So we think it could be a potential beneficial effect that you have translocation of annexin A2 to the cell surface after crystal damage or crystal attachment and injury. The third bullet, calcium oxalate induces mitochondrial function, apoptosis, ferroptosis in these MIMCD3 cells. Next, calcium oxalate increases the density of lipid rat, which is known to activate piezo-1 activity in these cells. And piezo-1 activation leads to itself. The activation of piezo-1 leads to mitochondrial dysfunction in these cells. Calcium oxalate increases the formation of the NLRP3 inflammasome. This is at the membrane of these MYMCD3 cells. And finally, engineered immunoliposomes may represent a therapeutic strategy to inhibit or in this preclinical model, we're going to do this in animals next, to inhibit calcium oxalate induced cell death. So with that, I'll take any questions. Yes.
I just have a couple of questions. First is, what concentration of calcium oxalate did you use and what did you use for your vehicle?
Yeah, so that's a very good question. I think she's asking that question because she knows that calcium oxalate is relatively unstable, right? And insoluble. That's true. So for the vehicle, we have to use the same solvent that we use to dissolve or put the calcium oxalate in solution. And that's a very mild acidic solution of citric acid and PBS, normal saline, but a little bit of citric acid. And the concentration of, to answer your second question, the concentration of calcium oxalate, it's been a while since I did these studies. I want to say it's, do you remember any, Harika?
It should be 1 micromole.
One micromole, right? Yeah, 1 micromole.
How close is this to the physiological concentration of the kidney tubules?
Yeah, that's a good question. So, the kidney tubules, the fluid flowing is dynamic, it's not static. And do you know, Dr. Khan, the concentration of calcium oxalate that
Are you kidding me? You saw my paper I published in 1999.
That's
No, but the question that you are asking is, I mean, he's talking about the luminal part. So there has to be very high concentration of oxalate there. And so and once they make the crystal, they basically stay there.
Right. So
I can just tell you it's hot. I can't tell you the,
it's pretty high, right? Yeah. So the question is, there direct toxicity due to high concentration or relatively easily precipitating molecule versus toxicity from it being oxalate by itself?
Again, I think the part is this: there is regular hyperoxaluria, which is very uncommon in idiopathic kidney stone formers. Then we have a primary hyperoxaluria, which is so many of them. And then there is enteric. So sometime, I don't know, I used to tell it when I was teaching, that if you eat an oxalate-rich diet, and pretty soon you go look at your urine, you may actually see calcium oxalate crystals. So it can happen that oxalate can increase very quickly But when the crystal deform or crystal, they just get out.
Thank you. Yes. Calcium salts, flow calcium salts are relatively insoluble, right?
Very inside.
With a few
Well, without the
Yeah.
mild acidic buffer, we can't dissolve it. We can't put it in solution.
That makes sense.
Yeah, another question. No, Dr. Shoemaker has a question. You want to ask your question?
This is pretty easy, but those intermedullary collecting duct cells, do they form confluent monolayer?
Yes, good question.
And
So-
then you're putting your substance on the intertubular layer or the outer?
Good question. So the question that Dr. Shoemaker had was that, do these IMCD3 cells express the medullary sensitive epithelial sodium channels in the luminal apical plasma membrane? Yes, and they do form a monolayer, so it's a type phenotype, and because of that type phenotype, we can measure transepithelial resistance, transepithelial voltage, and using Ohm's law, calculate transepithelial current. And yes, to answer your second question is that Okay, come back.
That was a perfect question, because I was asking about the same cells, cell culture as well.
Yeah.
They're human.
There's not. Unfortunately, there's not a human, otherwise we would use it. There's not a human. We can isolate principal cells. In fact, we can patch native tubules. So we're one of only a few labs in the world that can do that. You know, there's 1 million nephrons per kidney. We can microdissect one, fillet it open, and patch principal cells in the collecting duct. So if we can get some human biopsy samples, we can do that, and we can actually isolate and make a principal cell, intermedullary collecting duct cell line, but there's not one available. Good question, excellent question.
Is there a mouse model or is there an
Many
animal
mouse-
model of calcium?
There's many, and Dr. Khan published extensively on that, several review papers. Again, I just published a review paper that addresses some of the things I talked about today. But there are many rat and mouse models, yeah. And as I alluded to earlier, the stone formation is luminal in most of those animal models. Other questions?
I have a,
Yes,
sorry,
Please,
probably like a
please.
silly question.
no such thing.
I was just thinking, maybe, like Dr. Khan said, you eat an oxalate-rich diet and you urinate, you eat the oxalate. But not everyone who eats a high oxalate diet will make kidney stones.
Right.
And so if we want to invoke the same mechanisms, pathogenic mechanisms that you presented, like what is it that sets people who make kidney stones, oxalate kidney stones, different from the phenomena that, just the presence of.
Yeah, so that's a very good question. The question Dr. Ritchie had is what makes someone susceptible? Because not everyone that eats a high oxalate diet will develop calcium oxalate stones, right? So there's a lot of working pieces here. There's different forms of calciuria. And if you think of someone being calcemic, you would expect them to have more calcium in the blood, but that's not the case. They're actually normal. They're normal calcemic, but they're calciuric. And then one thing you have to consider are the three mechanisms, right? So it could be absorptive hypercalciuria, it could be resorptive, and it could be renal, proximal tubule deficiency and handling of calcium. So that's one thing to consider. There are so many moving parts to that question. In terms of, I don't think it's dependent on diet as much as some other molecular determinants. So one thing I showed in my initial slides, the background slide, very important, and we sometimes overlook that. And that is the point I made or tried to make about a cell that is injured is more susceptible to having calcium oxalate being attached to it. So there's priming going on. So a second hit hypothesis is what I'm thinking about with. So if you have a cell that sees one stimulus or one hit, and then you have, diet could be the second, high oxalate diet, that would be enough to make those people more susceptible. But based on some studies that have been done, that I alluded to earlier, these intermedullary collecting duct cells are pretty resistant. Unlike the proximal tubule cells or other cell types that people have used. So, we're not talking about glomerular, podocytes or mesangial cells or endothelial cells here. Although people have used those, calcium stones don't stick to them. They don't originate from them and they don't stick to them very well. So, that's why we use this particular cell type. But that study showed, using the same cell type, that when cells are predisposed, when they're already injured, they're more predisposed to injury than if they were healthy.
And I think the presence of stone inhibitors in the urine prevents anemia.
Yeah, absolutely. And then you have to consider other etiologies and pathophysiologies like comorbidities, right? So CKD, hypertension, one of the things that we're doing. I like that question because it gives me an opportunity to talk about another project that we're doing related to kidney stones, looking at patients with different demographics, on different drugs, different comorbidities. So we have a large, working with Dr. Shatari, Dr. Kazori, Dr. Bird, Dr. Rucci. We're looking at patients that are hypertensive, patients that are hypertensive with CKD, with or without CKD. And we actually have some beautiful data in which we isolate urinary extracellular vesicles, and we take the stone specimens and we look at the matrix, and maybe I'll give a ground rounds on that soon, but that's a very good point, and I think it's a second hit hypothesis that's involved there. Other questions? A lot of questions. So more the better. We still have 5 minutes.
Very much fun.
Yeah. Thank you.
So, in diabetes, right, we know there's a significant mitochondrial dysfunction, in multiple organs, and reduced respiration, mitochondrial respiration.
Yes.
And so the question is that something, and all patients with diabetes are Dr. Uchi, you can correct me here, are more likely to have oxalate kidney stones compared to patients without diabetes.
So I asked her that same question a couple of days ago. We had a big discussion with Dr. Bird and Dr. Rucci. So if you read the literature, you're going to be very confused because there's some studies that were not done so well with AKI and CKD and diabetic nephropathy, diabetic kidney disease. So patients, and I think Dr. Rucci talked about this in her renal burn round, patients that have CKD actually protected against getting calcium oxalate stones. And we're actually doing that study with the diabetic kidney disease patients. That's one of our groups. So Dr. Shatari, Dr. Rucci, they're gathering enough, I want 100 patients, no less than 100, and they're getting me those patients. And Dr. Bird is getting me the stone specimens so I can analyze the matrix. All this we're doing right around the corner in my lab. So Niharika back there is isolating the urinary EVs from these patients because, it's really difficult to understand how after all these years of studying kidney stones, we don't even have a good assay to measure oxalate in the urine. There's no biomarkers. There are no good reliable biomarkers for calcium oxalate, calcium phosphate stones. So we're establishing a panel of novel biomarkers, including bioactive lipids and various proteins in the urinary EVs, and also looking at the matrix. So yes, in terms of diabetic kidney disease and CKD and AKI, there is literature on that. But you got to be careful because some of the studies were not done so well.
We don't, I'm not talking about CKD. We may or may not have
Hypertension too, I forgot hypertension.
I would wonder, right, if you're talking about mitochondria and diabetes, would there be an additive effect?
I think so, because you already have the mitochondrial dysfunction, and I think that's the first hit. So I think those patients are more susceptible to developing recurrent kidney stone formation. And that's where our data is showing us, actually. But I'm not gonna make any conclusions until I have at least 100 patients in each group. You know, so far we got like, what, 50 in each group? I want 100 to make that claim. Okay, well, thank you all for your.
Thank you very much.
Thank you. Thanks for coming.
I want to ask, unless I wanted to say.
College.
He wasn't here. Now he's going to send the tape.
Yeah, exactly. Being metabolized here. The membranes. She's doing it. Bees that we're seeing from the patient. Really nice. Yeah, really nice. Human stuff. Coming to get really nice. Human urine from your patients. Sorry.
I don't know how it's.
And
3 kids.
in addition to that, these lipids are vital. So they're blood-brain.
Yeah, I think she was, I don't think. Yes.
Look at rafts, it's causing mitochondrial dysfunction. Not just take a look at your Thanks for being part of it.
Yeah, but I have.
I'm gonna take you, this is the nurse maker.
Okay, I'll send it.
Okay, we don't work with the sound, but I want to ask a very simple, probably question. And that is, do patients actually with this who have both uric acid erythrocytosis and calcium blood supply? I mean, do you have both in one patient?
I mean, not even conditions, like, that kind of thing. But one thing I can tell you, that uric acid can become cycles.
I mean, that means it's not just many laxity or the lungs. I mean, it can be.
That's calcium.
Uric acid. Crystallization.
But in my limited practice, I've never seen anybody, I mean, the only animal that makes gas or something like
Thursday.
So I was wondering, would there be any, personally, I've never heard of a patient who had both. And is that logically important?
Perfect.
Because if you have uric acid, I just uric acid and